Hello students, today we'll be studying about the second week of development, that is the development of the bilaminar germ disc formation. Now we'll be studying about the cleavage and the formation of a blastocyst. We have already studied about the cleavage and the formation of blastocyst during the first week. The development of the trophoblast, the process of implantation was partly studied in the last class. The describe the formation of extra embryonic mesoderm, extra embryonic selum, the bilaminar disc, and the procaudal plate. Then you should be able to give a brief account of abortion, the decidual reaction, and the pregnancy test. So firstly, during the second week, the implantation it is about to complete. The blastocyst it sinks in the endometrium due to the invading capacity of the trophoblast. So the implantation gets completed by the 12th day. Now there are certain developmental changes in the second week. First are the changes in the trophoblast, then is the changes in the embryoblast, and finally the changes in the endometrium. So firstly, at the day 8 of implantation, you can see this one is the blastocele, this one is a blastocyst, this one is a blastocele, this one is the appearance of the amniotic cavity, this is the epiblastic cell and the hypoblast cell of the inner cell mass. This one is the outer cell mass which is organized itself into syncytial trophoblast and the cytotrophoblast. Now, what are the changes in the trophoblasts? So by the day 8, the blastocyst it gets partially embedded in the maternal endometrium. The trophoblast it gets differentiated into the inner cytotrophoblast cell layer and the outer multinucleated with the diminishing cell membranes. They are known as the syncytial trophoblast. So cytotrophoblast is only giving rise to the syncytial trophoblast. Then what are the changes in the inner cell mass or the embryoblast? It gets divided into or organized into the two cell layers. The cuboidal cell layer, which is the hypoblast, which is facing the blastocele, and the flattened cell layer, that is the epiblast, which will face towards the embryonic pole. Later on, what happens is this is by the day 9, it has completely embedded in the maternal endometrium and the defect has been closed by the coagulation plug. Now, what happens? The epiblastic cells, they get organized into columnar cell layer and they will start because of the compaction, there is an appearance of the amniotic cavity amongst the epiblast cells. So there are some of the cells which is towards the embryonic pole, which is forming the roof of this amniotic cavity will form the embrioblasts, and the columnar cell layer, it will form the uh, floor of the amniotic cavity. Now, this is the hypoblast cell layer. So this is facing the blastocele seal. Now this is the formation of the bilaminar germ disc, which marks the formation of the dorsoventral axis of the embryo. So this is the day 9. Now there is a formation of trophoblastic lacunae. This one is the outer cell mass, which is the trophoblast. Fine. So this is the cytotrophoblast. This is the syncytiotrophoblast, and you can see the lacunae. They have appeared in the syncytiotrophoblast. The defect is closed by the fibrin plug, and that is by the 28th day. That corresponds to the 28th day of the menstrual cycle. So it might be confused by the normal menstrual bleeding. Now, what is the changes in the trophoblast? The blastocyst, it further penetrates the endometrium. The penetration defect, as I have told you, it is sealed by the fibrin plug. The syncytiotrophoblast, it develops more towards the embryonic pole. Now, the cells of the syncytiotrophoblast grow rapidly and they lose their adjacent cell membrane to form a multinucleated protoplasmic mass. Now, there is a lacuna stage in the trophoblast. A small lacuna, they develops into the syncytiotrophoblast. So these lacunar spaces fuse with each other to form large lacunar spaces. Now what are the changes in the embryoblast? During the day 9 to 10, there is a formation of exosalomix Huesner's membrane. The cells of the hypoblast, they will proliferate and they will line the blastocele, and then it will convert the blastocele into the primary yolk sac. And this layer is known as Huesner's membrane. So now this cavity, the blastocele, it gets converted into the primary yolk sac or the exocelomic cavity. So where were we? We missed it. Yes. So next is day 10 to day 11. Now this marks the formation of the extra embryonic mesoderm. Now this brown one you can see, this is intervening between the Huesner's membrane and the cytotrophoblastic cell membrane. So this is the extra embryonic mesoderm, which is formed by the proliferation of the hypoblastic cell layer. So hypoblastic cell layer is the only cell layer which is giving rise to the Huesner's membrane as well as the extra embryonic mesoderm. Else we have this hypoblast cell layer has no other work to do in an embryo. The epiblast cell layer is said to be the mother of all the cell layers of a developing embryo. So now over here you can see what are the other changes which you can see that the lacunar spaces have developed in the syncytial trophoblast. They are filled with the blood. So the syncytial trophoblast, they have already invaded the maternal capillaries. So the lacuna, they get filled with the blood. And over here the lacuna also starts appearing into the extra embryonic mesoderm at the expense of the primary yolk sac membrane. Primary yolk sac, some of the primary yolk sac, it has been pinched into the extra embryonic coelomic cavity. So over here, what are the changes in the trophoblast? As I've already told you, the lacunar spaces, they are enlarging and they form communications with each other. And this change is more prominent along the embryonic pole. Now the cords of the syncytial trophoblast, they will form the trabeculae. Now the syncytial trophoblast, they will irrate the maternal capillaries and other vessels, and hence the lacunar spaces, they get filled with the maternal blood. So erosion of the maternal veins, they will appear early, that will take place earlier than the maternal arteries. Now the filling of the lacunar spaces with the maternal blood establishes uteroplacental circulation. So uteroplacental circulation is established at 11th to 12th day of the intrauterine life, and it is by the invasion of the syncytial trophoblast uh, to the maternal cap, that is the maternal capillaries, they are invaded by the syncytial trophoblasts. Now what is the changes in the embryoblast? The cells of the yolk sac, they will start multiplying and they will form the extra embryonic mesoderm. Now this extra embryonic mesoderm, again it will start spreading and it will separate the germ disc with the amniotic cavity and the yolk sac from the cytotrophoblast. So extra embryonic mesoderm, it will intervene between the cytotrophoblast and the amniotic cavity and the yolk sac. Now the cavities will start appearing in the growing extra embryonic mesoderm. These cavities soon they will fuse with each other and it will lead to the formation of extra embryonic coelom or the chorionic cavity. So the extra embryonic coelom, it will isolate the yolk sac and the amniotic cavity from the cytotrophoblast at all the places except at the caudal end of the germ disc, where it forms the connecting stalk of extra embryonic mesoderm. So connecting stalk will ultimately form the umbilical cord. Now, the exocelomic cavity, it divides the extraembryonic mesoderm into the splanchnopluric mesoderm, which is facing towards the secondary yolk sac, and the somatopluric mesoderm, which is facing towards the cytotrophoblast cells. Now, this exocelomic cavity or the chorionic cavity, when it will appear, it will automatically squeeze the primary yolk sac, which will be called as the secondary yolk sac. So we have two mesoderm, the somatopluric mesoderm outside and somatosplanchnopluric mesoderm, which is towards the secondary yolk sac. So over here you can see this is the chorionic cavity. This is the primary yolk sac. This is the future connecting stalk. Still this chorionic cavity has not encircled the growing embryo. This is by the day 12 and 13. And though the uteroplacental circulation has been established. Now, what is the chorion, the trophoblast, and the somatopluric? This is the trophoblast and the somatopluric layer, which includes the extraembryonic mesoderm, the somatopluric mesoderm. So, the trophoblast and the somatopluric mesoderm, the extraembryonic mesoderm together forms the chorion, and the blastocystic cavity will now be known as the chorionic cavity. So, this will grow in size gradually, and this cavity will finally be reduced. The yolk sac will be reducing. So again, here this is the exocelomic cyst. Sometimes a cyst remains and it is not fused. It is being pinched off, though it is being pinched off from the yolk sac, but it is not totally incorporated into the chorionic cavity. So it might divide the chorionic cavity into the two. So that is the exocelomic cyst. So finally, now what will be the changes in the trophoblast by the day 13 to 14? So the syncytial trophoblast, they will form the villi. They will form the columns like fingers, and that is known as villi. So the outer boundaries of this villi is formed by the syncytial trophoblast, and the inner core is formed by the cytotrophoblast. So this is the primary stem villi with the central core of the cytotrophoblast, and the peripheral part forms by the syncytial trophoblast. And between the two villi will be the trophoblastic lacunae filled with the maternal blood. Now, what is the changes in the embryoblast? There is a formation of the secondary yolk sac, and which is formed by the portion of the primary yolk sac. It is being pinched off by the developing chorionic cavity. It will result in a formation of a smaller secondary yolk sac, and some of the cells of the hypoblast, they will get elongated. They will become columnar, and they will mark the formation of the precaudal plate, which will marks the cephalic end of the embryo, and the rear end will be the caudal end. So it marks the cephalocaudal axis of the embryo. So what is the significance of the precaudal plate? It shows the first sign of differentiation. It establishes cephalocaudal axis, and it is an important organizer of the head region. So what is an organizer? There is certain regions of the embryo which has ability to influence the differentiation of neighboring regions. So the influence which is exerted by the area is called induction, whereas the area exerting induction is called organizer. In interactions between tissues, one is inductor and the other one is the responder. So capacity to respond to the inductor is called competence, and the factors that influence the competence to respond are called competence factors. So fetus as graft. Now, the transfer of tissue from one member of species to another member of species in human is known as allograft. So it is between the same species. And autograft is this amongst the same person. That is, excuse me, from one body space to from one body part to other. So mother generally accepts the fetus. It is not rejecting it, and without inducing any immune reaction. So what is the reason behind it? So there are 4 reasons which was given by Medover in 1953. That is the physical separation by the trophoblast which poorly expresses major histocompatibility complex, which are certain receptors coated by each and every cell of the body. So trophoblast poorly expresses MHC molecules, and this is the layer which is separating the fetus from the mother. So which is essential to generate an immune response. Then the maternal system is tolerant to the conceptus. Why? Because specific hormonal conditions they make maternal immune system non-responsive to the fetal tissue. Then antigenically immature conceptus. The conceptus is antigenically immature. And though some of the maternal T cells, if they are activated, they undergo programmed cell death, which is known as apoptosis, under the certain factors, which is the tumor necrotic factors. There is a condition which is known as genomic imprinting. So, in which the DNA of female germline is more methylated than DNA of the male germlines. So, the basis of genomic imprinting is methylation of DNA that leads to gene silencing. Two important example of genomic imprinting is the deletion of long arm of chromosome 15, which leads to the Prader-Willi syndrome if it is inherited from father, and if it is from the mother, then it is known as Angelman syndrome. Then what is lionization? There is inactivation of an extra chromosome in the females. So Dr. excuse me, Dr. Mary Lyon proposed that one of the two X chromosome in normal female is functional and the other is condensed and inactive in early developmental period. It is called bar body or sex chromatin. This condensed chromosome can be paternal or maternal in origin. Thereafter, the same X chromosome is inactivated in all the daughter cells. This process of inactivation of X chromosome is called lionization. So this is just a summary of what we have uh, studied today. That is, firstly, there is a modula which by repeated cleavage and by undergoing compaction, there is an appearance of a cavity which is known as blastocele. So now it is known as a blastocyst, and it has already been hatched at the 6th day. That is, zona pellucida has been disappeared, and it will now start invading the maternal endometrium. Now the if you remember, maternal endometrium was at the secretory stage. So whatever this changes which is occurring in the maternal endometrium, it will result in increased secretion by the maternal endometrium, and this is known as the decidual reaction. Now, again, there is the ectoderm or the epiblast cell, they will become columnar, and the endodermal or the hypoplastic cells, they are cuboidal. Then there is appearance of the amniotic cavity, and there is formation of amniogenic cells. There is appearance of the this primary yolk sac. When this hypoblast cell layer, it will proliferate and it will form the Huesner's membrane. Then after that, the hypoplastic cell layer, again it will proliferate, it will form extraembryonic mesoderm, it will convert the blastocele into a primary yolk sac. Then finally there is appearance of lacunae in the inner this extraembryonic mesoderm, which will divide this extraembryonic mesoderm into the splanchnopluric and the somatopluric mesoderm. Then there is elongation of certain hypoplastic cells, which is known as precaudal plate, which will demarcate the cephalocaudal, or the which will establish rather the cephalocaudal axis of an embryo. So this is the precaudal plate, this which is denoting the head end and the tail end of an developing embryo. So why the second week is known as the week of tooth? Because the embryo, it divides into an inner and an outer cell mass. Inner cell mass known as embryoblast. Outer cell mass is known as trophoblast. The inner cell mass differentiates into hypoblast and the epiblast. That is the endoderm and the ectoderm. The appearance of the two cavities, the amniotic and the yolk, amniotic cavity and the yolk sac cavity. Then there are two layers of the trophoblast. That is, the trophoblast divides into the syncytial trophoblast and the cytotrophoblast. Then This hypoblast cell layer will give rise to extraembryonic mesoderm, which will form the somatopluric and the splanchnopluric mesoderm by the appearance of the chorionic cavity. Now, there are two fetal membranes. One is the amnion, another one is the chorion. Chorion is the somatopluric mesoderm along with the trophoblast, and the amnion is the splanchnopluric mesoderm along with the embryogenic cells. Then there are two ends of the embryo which is established because of the precaudal plate, that is the cephalic and the caudal end, and the two axes, the dorsal and the ventral axis. So finally, this is the summary. That is, after the morula, it will undergo cleavage, that is the mitotic division. Then under the compaction, it is getting divided into outer cell mass, that is trophoblast, and the inner cell mass, which is embryoblast. The outer cell mass again divides into the cytotrophoblast, which will give rise to the syncytial trophoblast. So two cell layer are formed over here. Now the inner cell mass divided into the epiblast and the hypoblast cell layer. Hypoblast cell layer will give rise to the Huysner's membrane, and the epiblast cell layer, it is the mother of all the other cell layer except the extraembryonic mesoderm, which is derived from the extraembryonic endoderm or the hypoblast cell layer. That's all.